So welcome back again. Let's take a look at our concept check questions, and then we'll talk about Mendel's second law. If two organisms have the same genotype, must they have the same phenotype? The answer to that is yes, because irrespective of the genotype, if they are the same, then the phenotypes will be the same, because remember, the phenotype is the product of the genotype. So if they have the same genotype, say they're homozygous dominant, then they must show the dominant trait. If they're homozygous recessive, they must show the recessive trait. If they're heterozygous, they must show the dominant trait again. So if two individuals are homozygous dominant, then they're going to show the dominant trait. If they're heterozygous, they're still going to show the dominant trait. So if they're both heterozygous, then they must both show the dominant trait. If they are both homozygous recessive, then they must show the recessive trait. However, if they have the same phenotype, must they have the same genotype? The answer there is no. If they have the recessive phenotype, then they must have the same genotype, the homozygous recessive. But if they show the dominant phenotype, they could be homozygous for it, or they could be heterozygous, because both of those individuals will show the dominant phenotype. So just because they're showing the same phenotype does not imply that they're going to have the same genotype. If they have the same genotype, then they must have the same phenotype. Gets a little bit confusing, but that's the way it is. Mendel showed that all organisms have two alleles for every trait. Where do these alleles come from? That's what we were talking about just before the break. Those alleles obviously have to come from our parents. One allele comes from each of the parents via the homologous chromosomes that we inherit from each of our parents. So that's where they come from. So Mendel's other law, called the law of independent assortment, is based on dihybrid crosses. So here he's looking, or he's starting with individuals that have, or that are pure breeding for two traits of two characters, hence the term dihybrid. So he started with pure breeding parents that were pure breeding for two dominant traits. Let's get the pen back up again. So the way he set this up was not arbitrary. Again, Mendel never did anything arbitrarily. So he had pure breeding parents that were pure breeding for the two dominant traits of the two characters he was looking at. And he had parents that were pure breeding for the two recessive traits of the two characteristics that he was looking at. So basically, this is an extension of what he was doing with the monohybrid cross. And the question that he wanted to know was that if you start out with parents that contain the two alleles for the dominant trait, or the dominant traits, would those continue to be inherited together in subsequent generations? And if you started out with parents that contain the two alleles for the recessive traits, would those be inherited together in subsequent generations? Or was it possible that the alleles for the two separate traits could be separated in any possible combinations? What he really needed was not these pure breeding parents, but parents that were heterozygous for these traits, and those he could get from the F1s. So when he started out with his pure breeding parents, The idea of having these pure breeding parents was so that he could get F1s that had combinations of the two alleles. Again, an extension of what he'd already found in his monohybrid crosses. Remember, the F1s there were heterozygous individuals, starting with his pure breeding parents. So he knew at that point he had combinations of alleles. So when he starts out with his dihybrid crosses, he starts out with his pure breeding parents again, with either the two dominant traits or the two recessive traits. Breeding those together would then give him his heterozygotes, and they would all show the dominant traits that he was following. Then he knew he needed to go on to the F2 generation, so he could allow his F1s to either self-pollinate or transfer pollen between two of them, irrespective of which way he did it. He knew he was going to be generating his F2s, and he, of course, would go ahead and do it both ways because that was the sort of person that he was. If the combination of alleles in the original pure breeding parents is retained through the F1s and on into the F2s, then the resulting offspring are simply going to show a 3-to-1 ratio. And we can illustrate this in this diagram. And the reason why is because this individual, as a pure breeding parent, can only give you a combination of the big R and the big Y. Now, you can't look at this and say, well, it could pass on the two big R's. No, it can't. It can only pass on one of the R's and one of the Y's. Remember, that's what he'd already discovered in the monohybrid crosses. You don't pass on both alleles. You only ever pass on one of the two alleles. And if you really want to know what's being represented here, the big R is actually one homologous pair of chromosomes, and the big Y is another pair of homologous chromosomes, or potentially could be. That's one way of interpreting it. Mendel didn't know whether that was the case, but that's what it would actually turn out to be. But the only combination of R and Y that you could get from this individual is the big R, big Y, and the only combination of R and Y that you can get from this individual is little r, little y. So here are F1s. If the RY combinations are retained as they were in the original parental, then the only possible combination in the F2 would be big R, big Y, and little r, little y, which would be the same in the second parent. And when you combine these together, what you get is, I think I'll do this in black so it stands out a little bit better. Big R, big Y, big Y. This would be big R, big Y, little y. This would be big R, big Y, little y. And this would be little r, little y, little y. And because big R and big Y are dominant, yeah, maybe I should do this in purple. That would be round and yellow. That would be round and yellow. That would be round and yellow. And this would be wrinkled and green. And so you would get your 3-to-1 ratio. If the big R, big Y, and the little r, little y combination are the only possible combination in the alleles, the ones that you started with are the ones that you continue to get in subsequent generations. If, on the other hand, you could mix them up, so you could still get the big R, big Y and the little r, little y combination. But then there are two other possible combinations that you could get. You could get the little r with the big Y, and you could get the big R with the little y. So there are 4 possible combinations from each of the parents. That means you get a Punnett square of 16 squares, and these are all the possible combinations. You get 9 big R, big Y, in other words, round and yellow. You get 3 little r, little r, oops, let's get rid of that. Actually, let's get rid of all of that and do it this way. You get 3 of these. which is the big R with the little r with the big Y, so wrinkled and yellow. You get, well, green won't work on that one, will it? Let's do it in black. We get 3 of these, which is the round, which is the dominant, with the recessive green color. And then you get, let's do blue, I guess, one of those, which is the wrinkled and green, the double recessive. So you get 9 double dominant, 3 dominant, 1 dominant and recessive like this, another 3 with the flipped dominant and recessive, and then 1 double recessive. And the ratio is 9 to 3 to 3 to 1. And this is the result that Mendel generated. We actually have his numbers for this one as well. So we know that this is the result that he's got. So it is possible to get all possible combinations. That means if we're thinking in terms of what we're referring to up here, big R, and big Y, and the little r, and little y, oops. And you know what I'm going to put here? Homologous chromosome. So when we start thinking about the fact that these might represent pairs of homologous chromosomes, which is in fact what this data is telling us, how does that in fact explain all of these possible combinations? And the answer is because of the independent and random behavior of the chromosomes, the homologous pairs of chromosomes during meiosis. And if that description, random and independent behavior, starts to ring a few bells from last week, then good, because it should, because that independent and random behavior is the way the chromosomes, the pairs of homologous chromosomes line up in metaphase 1 on the metaphase plate. This is the origin of Mendel's law of independent assortment. Basically what he's saying here is, in modern terminology, each pair of homologous chromosomes sorts itself out or separates randomly and independently of what each other pair of chromosomes does. And that behavior is specifically in metaphase 1, when each pair of homologous chromosomes lines up on the metaphase plate randomly. Remember, they could be one way or the other way, they could be flipped either way. And what one pair of chromosomes does is independent of what all the other pairs of chromosomes do. And that's what leads you to all the possible combinations. And so what Mendel has discovered here is, again, a behavior of chromosomes in meiosis. At least that's the way we interpret it. And we know that that's in fact correct. So the interpretation is not inaccurate. I mean, it's actually quite accurate. So the example that's given in the textbook is, unfortunately, is somewhat of an unfortunate one. But it reflects, again, breeding of dogs. And in this case, an unfortunate result of poor breeding choices in the past, because it turns out that Labradors have a mutation that is that results in loss of vision. The dogs are unfortunately born blind. And if we look at it in combination with coat color, we know the location of the coat color gene, and we know the location of the vision gene. They happen to be on separate pairs of homologous chromosomes. So they are going to be inherited in a Mendelian fashion. So if you start out with parents that are phenotypically normal, in other words, they have black coat color and normal vision. So they're phenotypically normal for black and normal coat color and normal vision. Their litter is going to reflect Mendel's law of independent assortment. There will be 9 that have the same parental phenotypes, black coat color and normal vision. There will be 3 dogs that are black but blind. So remember, black is the dominant coat color. Blind is the recessive, fortunately, vision characteristic. So it's the dominant black with the recessive blind. So that's the dominant versus dominant recessive, one dominant recessive combination. Then in terms of coat color, chocolate is considered to be the recessive coat color, and normal vision is the dominant, fortunately, vision trait. And so here's the other dominant recessive combination. Normal vision, the dominant trait, and chocolate coat color, the recessive coat color. And then unfortunately, one individual is going to show the double recessive, chocolate and blind. So it does actually follow Mendel's patterns of inheritance. We have many examples of how this will work. This happens to be the one that the book has chosen to create that connection back to our ongoing theme of dog characteristics and dog breeding. So there is one more genetics concept that we need to talk about as far as Mendelian genetics is concerned before we can go on and talk about other inheritance patterns that are related to Mendel's observations, but not specifically Mendelian. And that is the notion of what we refer to in modern genetics as a test cross. Why would we need a test cross? Well, what Mendel had established in all of his protocols for doing genetics experiments was the requirement for knowing exactly what the genotype of your starting parents is. That's why he worked really hard with his assistants for 2 years to create the pure breeding parents that he needed to begin his experiments. Because if you want to understand what you get at the end of a genetics experiment, you need to know exactly what you're beginning with. In other words, you need to know the genotype of your parents in order to be able to figure out what the genotypes of your offspring are. If you don't know the genotypes of your parents, of the parents in your genetics experiment, then you have no way of understanding why you got the result that you got at the end, because you simply can't track it through. The problem is, as we have seen, if you have an individual who shows the dominant phenotype, you don't really know what their genotype is. They could be homozygous for it, but they could be just as easily heterozygous. In fact, the only phenotype that you ever truly know with absolute certainty what its genotype is, just by looking, are the individuals who show the recessive trait, because they have to be homozygous for it. But that is the basis for doing the test cross. Because in a test cross, you take the individual whose genotype you don't know, that is the individual showing the dominant phenotype, but you have no clue what their genotype is, and you cross them with an individual showing the recessive trait. In other words, you know exactly what their genotype is. Why would that be the case? Well, if we look at it in terms of Mendel's flower color, an individual that has the dominant trait, the purple trait, could be big P, little p, or they could be big P, big P. put the tail on the little p so that we can identify which one. Our partner in this cross is going to be individuals that are little p, little p. And because this individual can only contribute the little p allele, and that's an absolute. Any difference that we're going to see in the offspring must be as a consequence of the purple parent being homozygous or heterozygous. So if the individual is homozygous, actually what I'm going to do is this. Excuse me. So we'll do it this way. So if the parent is big P, then what you're going to wind up with is individuals who are this combination, and they're all going to be purple. If, on the other hand, move this box out of the way, the individuals in big P, little p, then you're going to get a big P with a little p, or you're going to get a little p with a little p. And so what you're going to get is 1-to-1 purple to white. Again, the reason why this works is because the individual who shows the recessive phenotype is homozygous for it and can only pass on one particular allele, in this case, the little p allele or the white allele. If the parent showing the dominant trait is homozygous for that, then all the offspring are going to be big P, little p. This is basically the parental cross in Mendel's monohybrid cross. You get every individual who's going to be heterozygous. That means you're going to get all purple. If, on the other hand, the unknown parent, the one with the purple trait in this case, is heterozygous, then they could pass on a big P, but they could just as easily pass on a little p, which means you're going to get a big P, little p combination, or a little p combination, a 1-to-1 purple to white. And so you can easily tell from the test cross what the genotype of your unknown is on the basis of what you see in the offspring. Because if they're all purple, you know that the original parent was homozygous. If they're half purple, half white, you know the original parent was heterozygous. It's pretty straightforward. And the book again features the dogs with coat color in this case, black versus chocolate being the recessive, so that they're homozygous for that. We've got our black, could be heterozygous, could be homozygous, and you can see the outcome here. If the black parent is in fact homozygous, then all the offspring will be black. If the black parent is heterozygous, then you're going to get half black, half chocolate, half dominant, half recessive. It's a fairly straightforward test, and we still use them today. Test crosses are used routinely, and they have, in fact, an interesting history in genetics research. We don't get into that in this chapter or in this class, but there's some interesting experiments that were done with Drosophila that involved test crosses that gave us some interesting results and led to a new understanding of genetics patterns. So test crosses are actually quite important. The other thing that we don't dig very deeply into in this class, but which you ought to be aware of, is that the reason why Mendel was able to figure things out and interpret his data was because he understood probability. In fact, inheritance is really all about probability. They're just probability problems. And it's no different than the probability of tossing a head or a tail on a coin. Because if you want to know the connection, the genetics equivalent of a coin is a heterozygous individual, right? Because a heterozygous individual has two different alleles, the allele for the dominant trait, the allele for the recessive trait. A coin has a head and a tail, two different sides. And so if you understand the probabilities of tossing coins, which is very easy to understand, then you can solve genetics problems very easily, and you can solve what looks like really difficult genetics problems quite easily by understanding the notion of probabilities and what Mendel's laws tell us about the behavior of pairs of homologous chromosomes. So if we take a look at a simple Punnett square from the monohybrid cross of the F1 generation to give us the F2, we know that there are two different alleles. In this case, we're talking big B versus little b. But that, if we say that the big B is the equivalent of the head, the little b is the equivalent of the tail, which is what we typically do, then this is one parent. Here is the other parent, same deal again. Head is the big B, tail is the little b. The chance of tossing a head on any coin is 1 out of 2, or a half. The chance of tossing a tail on any coin is again 1 out of 2, or a half. So the chance of getting the big B on any of the from tossing any of these coins is 1 out of 2, just as getting a big B allele is 1 out of 2. Now, when you are dealing with what is referred to as dependent events, in other words, the outcome depends on two separate events, which is the case in this Punnett square. Getting the big B, big P is dependent on getting separately the big B from this parent and the big B from this parent. Those are two separate events that give you this outcome. So this, the big B, is dependent on getting the big B from here and the big B from here. Well, statistically, we apply what is referred to as the law of multiplication, which tells us multiply together the probability of getting the big B from here and the big B from here. Well, that's half by half, that's a quarter, which it turns out is the number of individuals that are big B in this Punnett square, 1 out of the 4. The same argument can be made for this individual. You need to get the little b from here, you need to get the little b from here. So getting the little b combination is dependent on getting the little b from here and the little b from here. So we multiply a half by half, that gives us a quarter, which is exactly the number of individuals that are little b, 1 out of the 4. Likewise for getting this individual, likewise for getting this individual. Now, there is a little bit more. This is not covered in the book, but I'm going to give you a little bit more. These individuals, the big B, little b, equals 1/2 of offspring. And there are two separate ways of getting there. They are independent of each other. They are totally separate. Getting this combination has no connection to getting this combination. How we get to this combination is very different from how we get to this combination, even though they are the same combination. There are two different ways of getting there. And because of that, we use the law of addition. Which means we add together these two separate. So a quarter plus a quarter, well, a quarter plus a quarter equals a half. And so the probabilities, the statistics come out. Again, these are half of the offspring. Each of them, this one is a quarter of the offspring, this one is a quarter of the offspring. These two together constitute half of the offspring. And the math actually works here. And if you are planning to take more advanced biology classes like our General Biology 1 class, then you will learn much more about solving genetics questions building on this basic concept. So for dependent events, so I just provide that same explanation for the big B combination. And I'll just put down here also works for little b. It's the same basic argument. So we don't dig any deeper into it than just this in this class, but as I said, if you're going to take more advanced biology classes like our General Biology 1 class, if that's your intention in the future, then we get into this in much more depth and we start looking at much larger combinations, but the basic math remains exactly the same, which is the beauty of the probability and the statistics approach to dealing with genetics problems, because that's really what they are all about. That was the underlying reason why Mendel was so successful in being able to interpret his data. We can simply adopt the same approach when it comes to answering more complicated genetics questions. So, time for another break. As I told you, I'm going to break this one up into many sections so that you have a chance to digest some of this, because there are some reasonably complicated ideas here. So here are our concept check questions for this break. You mate a chocolate lab with a black lab of unknown genotype for coat color. The resulting litter has only black puppies. Do you know the genotype for the black parent? Yes or no? And why? What is the probability of getting an ace in a 52 card deck of cards? What about an ace king pair? I've got a typo there. And what about two aces in a row? Can we predict those? I'll see you on the other side of the break with the answers.